In this lecture, we will be going over the anatomy and enzymology of fat absorption. Briefly, we will take a look at the anatomical components that are required for digestion and fat absorption, and we will then take a further look at the different chemical components that are involved, followed by the byproducts of fat digestion and where they will move into the body so that way they can be absorbed by the target tissues. So in this slide, we see a summary of every portion of the digestive tract and the associated mechanical and chemical digestive processes that are in each segment of the digestive tract, as well as the different portions that are involved in absorption. So we can see up here at the mouth that we have obviously mechanical digestion, chewing and swallowing are involved, as well as the chemical digestion of carbohydrates and a small portion digesting fats. This is typically achieved by the enzyme amylase to digest the carbohydrates, and the lingual lipase, one of the three lipases that are involved in fat digestion. Then as we move down the esophagus, we see here that we have the stomach. This is where we have peristaltic mixing and propulsion occur, as well as the chemical digestion of proteins and fats. This is where we will see a lot of our food gets emulsified with stomach acid as well. And we also start to see the absorption of lipid-soluble substances such as alcohol and aspirin. After we move out of the stomach, past the pyloric sphincter, into the small intestine, we then see further mechanical digestion taking place, including mixing and propulsion, primarily by segmentation and slow wave contractions by smooth muscle. And we also see chemical digestion of all types of nutrients. We have carbohydrates, fats, polypeptides, and nucleic acids all being broken down here by enzymes. Now, it is important to note that the small intestine is the site of the most chemical digestion, and almost all absorption occurs here. So chemical digestion breaks the large food molecules down into their component chemical building blocks. So we see proteins turn into amino acids, starches turn to carbohydrates, and so on down the chain. And a lot of the absorption occurs at the intestinal brush border. And we will get further into the other enzymes that are involved in fat absorption in the coming slides. So here in this slide, we have a more focused look at the anatomy of where fat digestion and absorption take place. Like we spoke about, we have the stomach, and just past the stomach, we have the pyloric sphincter. This is important, and this is important because it keeps the stomach acid where it belongs, in the stomach, obviously, and out of the small intestine where it could potentially do a great deal of damage. Other important anatomical structures to note, we have the liver, the gallbladder just underneath there. We also have, I believe we see the spleen right back here, and the pancreas. So up here in the top left, we have a more focused view specifically on the pancreas, the gallbladder, and the small intestine. Now, the gallbladder will come into play and is extremely important in fat digestion, as these large pools of hydrophobic fat pools will move out of the stomach in simply an emulsion and will not actually be broken down completely until they get into the small intestine. Another important thing to note, as we talked about, the small intestine is the site of the most chemical digestion and almost all of the absorption of nutrients, only source of lipase. So all of the lipase that you see, the lingual lipase, the gastric lipase, and the pancreatic lipase are all produced in the pancreas and are secreted from that organ. So it is very important, not just for diabetes or diseases of that sort, but it is also extremely important when it comes to fat digestion as well. So here in this slide, we have a view from our textbook in page 679 in chapter 21, or at least the first half of the image on that page. We actually see here how complicated a process fat digestion actually is. Because our bodies are technically aqueous environments, fat is not water soluble. It is hydrophobic, water fearing, as that would be said. And so in order for us to be able to digest these fats, we need a specific chemical ingredient, and that is bile salts. Now, bile salts are, they not only do they speed up lipid digestion, they're also essential to the absorption of the end products of lipid digestion. And not to be, they're not generally associated with the short-chain fatty acids. These are the fatty acids that are typically 10 to 12 carbons in length. Those short-chain fatty acids are relatively water-soluble and can enter the absorptive cells rather easily. But bile salts are polarized steroids that play a vital role in intestinal fat absorption. In the intestine, bile salts function as essential surfactants used to solubilize dietary fats in the hydrophilic milieu of the gut. Bile salts are synthesized in the hepatocytes of the liver from cholesterol. They're excreted into the bile and transported to the intestine via intra and extrahepatic bile ducts. In the bile and the gut, bile salts form a water-soluble aggregate known as micelles. Together with the fatty acids originating from the dietary fats, the formation of micelles are essential to the transport of the dietary fats towards the enterocytes across the aqueous intestinal lumen, an unstirred water layer as the intestinal epithelium. Absence of bile salts in the gut will result in severe intestinal fat malabsorption, and in recent years it has become clear that bile salts are not only involved in dietary food digestion, but also play vital roles in a variety of systemic metabolic regulatory processes. Now, micelles, these are large hydrophobic long-chain fatty acids, and monoacylglycerides are not so easily suspended in the watery intestinal chyme. Just like we had referred to, fats are hydrophobic. Now, a micelle is a formation of a tiny sphere, like we see down here, where we have the water-soluble heads and the water-insoluble tails forming these discs. Now, these discs are important because they allow for the fat to easily absorb along the brush border. Next, we're going to focus on the free fatty acids. The free fatty acids on monoacylglycerides that enter the epithelial cells are reincorporated into triglycerides. Triglycerides are mixed with phospholipids and cholesterol and surrounded with a protein coat. This new complex is called a chylomicron, and we will talk about that later on in this lecture. Here we have a slide discussing all the enzymes involved in fat absorption. Like we said, it is a relatively complicated process involving many steps and enzymes. And so, starting from top to bottom, we talked about lingual lipase starting to digest the fat in the mouth. We have gastric lipase, which is in the stomach. And we have the pancreatic lipase along with colipase. Now, This pancreatic lipase is a good enzyme, but it is not actually able to penetrate the bile salts that create the micelles. For that reason, it needs the colipase. And so what colipase does, it actually displaces the bile salts and allows the lipase to access the fat inside of the micelle. We see that phospholipids are digested by pancreatic phospholipase, and free cholesterol just diffuses simply because it is fat soluble and is able to cross over the cell membrane very easily. So finally, we have the steps of absorption. Now, absorption is important, as obviously we can see that not only do we have to break down our food, but we also have to take the nutrients from it. So here we mentioned chylomicrons. And we can see in this summary slide, we have all of the steps, again, from that same page from our textbook, all of the steps. And here we're going to focus on steps 4 and 5 over here. So we have the absorbed fats combined with cholesterol and proteins in the intestinal cells to form chylomicrons, and the chylomicrons are removed by the lymphatic system. Now, after being processed by the Golgi apparatus, chylomicrons are released from the cell, too big to pass through the basal membrane of the blood capillaries. Chylomicrons instead enter the large pores of lacteals. The lacteals come together to form the lymphatic vessels. The chylomicrons are transported in the lymphatic vessels and empty through the thoracic duct into the subclavian vein of the circulatory system. Once in the bloodstream, the enzyme lipoprotein lipase breaks down the triglycerides of the chylomicrons into free fatty acids and glycerol. These breakdown products then pass through the capillary walls to be used for energy by cells or stored in adipose tissue as fat. Liver cells combine the remaining chylomicron remnants with proteins, forming lipoproteins that transport cholesterol in the blood. And so we can see here that these chylomicrons, after they have been broken down, are then ready for use by the hepatocytes and other tissues of the body.